Good morning everyone. Good morning. Good morning all. Good morning. Just a moment, friends. Okay, very good morning all. So friends, we were doing transport in plants. So if every one of you very quickly recall what are the various concepts that we have finished so far in this particular plant physiology chapter. So we have completely done the photosynthesis. We have completely done the plant hormones, right? And we have also done secondary metabolites. In the secondary metabolites, friends, we are left with 7 to 8 slides. That is the cyanogenic glycosides. I will re-explain that. So, and we have completed the nitrogen metabolism, right? So now what we are doing, friends, of course we are doing the transport in plants. Okay, good morning. So friends, what we have to study while studying this particular transport in plants, I hope you have seen the February 2022 paper. We got one question in this particular transport from the matching question, right? Which is exclusively from the part C. You know, friends, whatever the question they have asked from this particular concept, surprisingly, that question will be basically from the NCRT level question they have asked. So that's why, especially in this topic, friends, we have to read a basic level concept also without fail. Okay, so friends, very quickly try to understand what are the various type of substances that can be transported into the plant body. We all people commonly know that water can be transported into the body, ions can be transported into the plant body, minerals can be transported into the plant body. What else, friends? Waters, ions, minerals, food can be transported into the plant body. So please listen me friends for 10-15 minutes continuously. Water, ions, minerals, food, all these has to be transported into the plant body. We all people know very well water can be transported into the plant body via xylem, whereas food can be transported into the plant body via phloem. We all people know about this. So friends, before understanding, listen to me very carefully everyone, before understanding how water can be transported in the xylem and food can be transported into the phloem, good morning everyone, first we have to understand, listen to me carefully, what are the different type of membrane transporters that are present on the plasma membrane, which we have to read compulsory. What are the various membrane transporters are there, friends? Channels are there, carriers are there, as well as, friends, pumps also there. See, I hope you are getting me correctly. Before reading the xylem transport, before reading the phloem transport, first we have to understand how does this particular ions are moving across the plasma membrane. How does the ionic movement occur, friends? Ionic movement occur with the help of a membrane transporters named as channels, carrier, pumps. So all the students, please kindly look into the diagram very carefully, friends. These are all what? Membrane transporters. And I want you people to remember one more important point, friends. all the membrane transporters are integral proteins. So friends, why I have used a word integral protein? Because friends, they are embedded inside the plasma membrane. Is it? You look into this, channels got embedded, carriers got embedded, as well as friends, pumps got embedded within the plasma membrane. So that's why friends, all the transporter proteins we generally call it as integral membrane proteins. Never ever forget it. So we just want to know what are channels, what are carriers, what are pumps, what are their examples, how they will work. So before going to the xylem and phloem transporter, we have to know this compulsory. Okay, focus for 15 minutes. Listen to me. First one, channels. So friends, the moment you heard the name of the channel, what is the most common point you have to remember? Everyone listen to me. Channels are gated like structure. What friends? Channels are gated like structure. The gates will be opened and closed, opens and closed with respect to the particular ligand. So you can understand friends, the ligand may be anything. The ligand may be neurotransmitter, the ligand may be growth factor, the ligand may be anything. What is channels actually? Channels are a gated like structure. Gates will be opened and closed with respect to the ligand. And what are the functions of this particular channels? Once they get opened, they will allow the movement of certain ions or molecules inside. And friends, here comes a very important question, that is, do you think every ion or every molecule can pass through the channel? No, not at all. I want every student has to understand. See friends, this particular channel having a particular pore-like structure. What friends? What the channel having? The channel having particular pore-like structure. Friends, every one of you listen. Whatever the ion or molecule can fit into this particular pore. Once again, listen to me. Whatever the ion or channel that can fix into this particular pore, only those ion or molecules only can be transported. So listen the statement what I am giving. Transport specificity is determined by the size of the pore. Never ever forget the point at all. What friends? Whatever the ion or molecule has to be transported, it will be transported based on the size of the pore. If this ion or molecule to be transported, if it fits into the pore, it can easily enter. If it does not fit into the pore, it cannot be transported. Kindly don't forget that. Now friends, what are the examples of this particular channels? The examples for these channels are anionic channel, calcium channel, potassium channel. And I want every student has to remember friends, whenever a particular, listen to me carefully, whenever a particular anion wants to go out of the cell, they will use the channels. So basically efflux channels. You are following me correctly, right? If the anions, say an example, chlorine, if it wants to go out of the cell, then only it will use the anionic channel. Suppose example friends, chlorine wants to enter inside, it never ever uses this particular channels. So for the efflux of chlorine or certain anions, they will use channels. And best example, calcium. If the calcium wants to enter inside, calcium enters inside the cell via calcium channels. I told you many times and once again I am repeating. Calcium is one of the major ion that plays a crucial role. In several physiological functions occurring inside the living cell. That's why friends, calcium will be required every single time in the living cell. Please kindly don't forget. Then friends, once you look into the potassium channels, you know, potassium channel wants to go out also, potassium channels wants to come inside also. They will use both influx and efflux of potassium can be mediated by the channels only. I mean to say, you can ask me, sir, when the potassium comes inside? Potassium comes inside when the positive charge is very low. When the potassium go out, when the potassium concentration is more than the normal, obviously friends, potassium is going out. So it's a very simple way. So potassium in and outward movement can be mediated by potassium channel, especially if the calcium wants to come only inside, what do we require? We require calcium channel. And the next one, anionic channel. If the anionic channel wants to enter inside, outside, especially chlorine, they will use this particular channels. Don't forget that. Next one, carriers. So friends, most of the students will think that carriers means we are greatly using the energy. No, carriers means we are not using the energy, friends. So it's basically use a mechanism which we all people know. We call it as the facilitated diffusion. Always remember that particular point. And listen to me very carefully, friends. All of you understand. This is a particular carrier, right? So friends, look into this. Certain ion or molecule cannot cross the plasma membrane. Now that particular ion or molecule which cannot cross the plasma membrane, they will be guided, they will be mediated, their movement can be facilitated. What friends? Their movement can be facilitated by a particular carrier molecule. You all the students try to look into this, friends. This is the ion which has to be transported across the membrane, but the point is here, they cannot cross the membrane. So that's why what happens, friends, this particular ion which has to be transported, it will come and associated at a particular binding site. Listen my words carefully. The ion or molecule which has to be transported, it will come and associated with the particular binding site on the carrier. Friends, the moment it will come and associated with the particular binding site of the carrier, immediately what happened, friends? Carrier undergo the conformational change. The carrier undergo the structural change. So because of the conformational change and because of the structural change, what happened, friends? Carrier will get opened. Very simple point. Now friends, once the carrier will get open, the ion or molecule basically enters inside. Very simple point, friends. Always remember, every carrier having a specific solute binding site. The moment a particular solute will come and associated with that particular binding site, immediately what will happen? It will undergo the conformational changes. The moment it will undergo the conformational changes, friends, it can easily allow the movement of that particular ion or molecule inside. Very simple point. Now I want you people to remember what kind of molecules or the substances that basically enter inside by using carriers. Mostly, friends, salts and amino acids. Mostly salts and amino acids, they enter inside the cell by using these particular carriers. We understand what are channels, we understand what are carriers. Remember, carriers are the type of transporters which always mediate the facilitated diffusion. Means there will be no requirement of the energy at all. It is just guiding, it is just facilitating, it is just mediating the movement of the ion or molecule. Simple point. So next one is what, friends? Next one we call it as the pumps. Friends, The moment you heard the name of a pump, all the student has to remember it is basically mediating the active transport. Okay, why we are using a word it is mediating the active transport? What is the reason? Because friends, whatever the ion or molecule that has to be transported, it is getting transported against the concentration gradient, against the electrochemical gradient. Once again understand, the movement of ion or molecule is what, friends? Against the concentration gradient. So if something is moving against, then obviously what happen, friends? We should use the abundant amount of the ATP. There is no doubt about it. Okay, now what are the type of pumps that can mediate the transport, friends? See, there are two type of transports are there: primary active transport, secondary active transport. Friends, every one of you don't get confused between the primary active and secondary active. One point is very clear: active transport means what, friends? Direct transfer, I mean to say, using the energy, that is for sure. But primary active transport means, friends, using the energy from the direct sources, using the energy from the direct sources, which we can call primary active transport. So directly using the ATP from ATP, directly using the energy from ATP, directly using the energy from NADPH, directly using the energy from oxidation and reduction reactions, whatever it may be, friends, we are using the energy from the direct sources. That's the point. Now friends, when you look into the secondary active transport, what is the statement I want you people to remember? Same, we are using the energy, there is no doubt about it, because we are moving the ion or molecule against the concentration gradient. So friends, but in the secondary active transport, the energy can be used from the indirect sources like H plus pumps, like H plus ATPase pumps. So that's why primary active transport, utilization of the energy from the direct sources; secondary active transport, utilization of the sources from the indirect sources. When we are reading the phloem transport, friends, automatically you will understand about the secondary active transport. Okay, every students, please kindly look into this. The primary active transport, it is also called as what transport? I told you, I want you to remember this, friends, because in a given 4-marker question we don't get the question directly from here, but in a given 4-marker question, as at least one statement we will get about the transport. So that's why we have to know it. Primary active transport, also called as a direct active transport. What did I tell you? It is using the energy from the direct sources such as ATP. Friends, what are the best examples of the pumps? Listen to me carefully. What are the best examples of the pumps that can mediate the active transport? Electrogenic pump, friends, followed by the electroneutral pump. Now you can ask me, sir, what is the differences between the electrogenic and electroneutral pump? Electroneutral pump means, friends, there will be a no net movement, right? So here there will be a net movement, there will be a no net movement, and my friends, here compulsory there will be a net movement. What's the difference? Let me tell you in a simple way. Suppose example, let's take an example of sodium potassium ATPase pumps. You all people know very well, friends. An example I am talking. Suppose 3 sodium is going out. If 3 sodium is going out, how many potassium is entering inside? 2 potassiums are entering inside. So friends, then there will be a net movement of 1 positive charge. Of course, true. 3 sodium are going out, 3 positive charges are going out. And how many positive charges are entering inside? Friends, only two positive charges are entering inside, which means friends there will be a net movement of one positive charge. Then what is electron neutral pump, friends? You can take an example that suppose one ion going out and one ion coming in, or 50 molecules going out, 50 molecules coming in, then there will be a no net movement. The net movement is zero. So don't confuse this electroneutral pump and friends this particular electrogenic pumps. These two are the two best examples of the pumps which basically mediate the primary active transport. Please kindly don't forget it. Next one, secondary active transport. I already told you friends, in the secondary active transport, the movement of the energy can be mediated by the indirect sources. We will discuss it in the upcoming slides, don't worry. The best examples of the secondary active transport pumps are uniporter, symporter, then friends followed by the antiporter. We all people commonly know very well what is uniporter. I have told you many times. Movement of the solute, movement of one solute, only one movement of one solute in one direction only. That is what exactly we can call the uniporter. Then movement of two solutes even in the same direction. And friends, please listen to me. Kindly don't think that symporter means only two solutes can come in same direction. Friends, you can remember in the by using symporter, three solutes also can come in the same direction. And friends, even by using the symporter, four solutes also can come, but all are moving what? All are moving same direction. So symporter most commonly is what? Two solutes. Most commonly. It may be three also, it may be four also. So symporter means movement of the solutes in the same direction. Antiporter we all know very well. Movement of the one ion inside, one ion outside. What you people can take in a best example? Sodium potassium ATPase pumps. And what you can take for the symporter? You can take many examples for the symporter. You can take sodium and glucose. In the human physiology also I told you, if the glucose wants to be transported, good morning, if the glucose wants to be transported in the body cells, it is compulsory. What it is compulsory? Glucose compulsory require the sodium. So friends, sodium and glucose are moving in the same direction. Kindly don't forget that point. Next one friends, H plus sucrose ATPase pumps. H plus sucrose ATPase pumps. So you know right, H plus is also moving inside. H plus only actually will guide, will mediate the movement of the sucrose inside. So H plus sucrose symporters, sodium glucose symporters. So friends, right now what we have studied, right now we just studied what kind of transporter molecules that are present on the plasma membrane which are responsible for the movement of the ions, like channels, carrier and pumps. Now exactly we have to study how does the water is moving, how does ions and minerals are moving through the xylem, how does food is getting transported through the phloem. This is the most important point where we can expect the questions. Before going to the xylem and phloem transport, I want every one of you to understand friends, another thing about the another transporter. What is that friends? Aquaporin. You know right? Aqua, water, porin, channels. Aquaporins are also called as the water channels. They are the channel proteins. They form a large family of intrinsic proteins. And friends, please listen my statements very carefully. Aquaporins are present in the plant cell also. Aquaporins are present in the animal cell also. But I want every student has to remember one of the common point. When compared to the animal cell, what friends? When compared to the animal cell, plant cell contain more aquaporins. Why friends? Because plant cannot move like animal, right? Plant cannot move like animal, so they have to consume more amount of the water when compared to the animal to control all their functions. That's why friends, more aquaporins are present in the plant cell. Kindly don't forget. And the point which were asked in the exam more than 4 to 5 times in the form of statements, that is what kind of ions can be transported through this particular aquaporins. I want every one of you to understand a point that aquaporins not only involved, please listen to me carefully, aquaporins not only involved in the transport of water, it is also involved in the transport of several uncharged molecules like urea. It is also involved in the movement of the uric acid, and it is involved in the movement of carbon dioxide, and it is involved in the movement of ammonia. This is the question they have asked about ammonia. Does the ammonia can cross the aquaporins or not? Of course friends, ammonia can easily cross this particular aquaporin, followed by the glycerol. Friends, all these ions, urea, uric acid, carbon dioxide, ammonia, glycerol, all can be transported via aquaporins along with the water molecule. Please kindly don't forget this. Okay, now friends, we are going to start reading briefly about the xylem transport as well as the phloem transport. We have to know about this very depth friends. Okay friends, whether from this particular slide onwards, I am requesting every one of you pay attention. If you get the question, we are going to get from here itself, that is the transport. How exactly the transport mechanism occurs, and what are the type of cells, or what are the type of proteins, or what are the type of transporters that are involved in this particular xylem and phloem. You all people know xylem is the one which can transport the water, ions and molecules. Phloem is the one which can transport the food. Before understanding xylem and phloem transport, the entire transport is depends on two pathways. One we called apoplastic pathway, and one we call it as a symplastic pathway. Friends, trust me, if you don't understand these two pathways, trust my words, if you don't understand these two pathways, you can't understand the process at all. You can't understand the mechanism at all. And anyway, let me tell you a simple word, symplast. Whenever you heard the name of a symplast, a common statement I want you to remember is that the movement can be exclusively mediated by what friends? Plasmodesmata. Kindly don't forget the statement. Then friends, what about the apoplast? Apoplast means friends, the movement can be exclusively mediated by the intercellular spaces. Okay, I will draw this. Just look into this, how actually the movement will occur. I am just drawing here how does the movement will be. Okay, all the students listen. What is apoplastic pathway? What is symplastic pathway? So friends, just listen to me. This is cortex. No. This is the epidermis. Then this is the cortex. What does the cortex is made up of? Cortex is made up of cortical cells. Cortex is made up of cortical cell. Then followed by friends endodermis. We all know very well endodermis having Casparian strip. Then finally xylem, right? This is the xylem. Now friends, every student try to understand what is symplastic pathway. I have told to all of you, through the plasmodesmata. Listen to me this very carefully. So friends, you know, right, plasmodesmata is the cytoplasmic bridge which is connecting, listen my statements carefully, plasmodesmata is connecting one cytoplasm of one cell to the cytoplasm of the another cell. Kindly don't forget it. So now look into this. See, one cell can be connected to the another cell via plasmodesmata. So friends, the movement is directly through the cell, inside the cell. This is plasmodesmata and this is the symplastic pathway. But friends, if you look into the apoplastic pathway, once again listen to me carefully. If you look into the apoplastic pathway, the movement will be like this. Listen to me. Through the cell wall, intracellular space. Through the cell wall, intracellular space. Through the cell wall, intracellular space. Through the cell wall, intracellular. Through the cell wall and intracellular. This is what movement? Apoplastic pathway. So I want every student has to look into this particular picture. You will get a little more clear idea. So you can see, no friends, all the students look into this. The arrow, whatever you are seeing here, the arrow, the red color arrow, whatever I am showing. So this is cell 1, this is cell 2. So through the plasmodesmata, ions or molecules are moving. Now whichever I am drawing with the blue color arrow, this is what pathway? Apoplastic pathway. Through the cell wall and the intercellular space. This is what pathway, friends? Apoplastic pathway. Never forget what is apoplastic and what is symplastic. You people commonly remember symplastic, plasmodesmata, and listen. Symplastic means through the cell, inside the cell. The apoplastic pathway, friends, whatever the ion or a molecule that is transporting by using apoplastic pathway, they never ever enter inside the cell. They never enter. They will only travel with the help of intracellular spaces only. Don't forget this. Okay. Now friends, I want every student has to understand this. You know, if you don't understand this slide, you can't understand the entire xylem transport. If you don't understand this, you can't understand the xylem transport. So pay utmost attention. I will explain twice, automatically you will understand. See friends, this is the root, correct na? If you do the cross section of a root like this, what we are getting? We will get the extension of the root hair. What do we call the extension of the root hair? Extension of the root hair called as epidermis. After epidermis, there is a cortex. After the cortex, there is a endodermis. Every one of us know very well the endodermis contain what, friends? Casparian strip. What strip? Casparian strip. Then followed by the pericycle, and friends, ultimately xylem. Is it? That is the point. We all people commonly know that. Now every one of you listen my words very, very carefully, friends. First of all, you can ask me a question. So this is the soil. Everyone please listen to me for 5 minutes. This is the soil. From the soil, how does the water molecules are reaching the root hair? That is the epidermis. How does the water molecule reaching to the epidermis? Of course, by the process of osmosis. Osmosis is the process where the water molecules are moving from the soil into the epidermis. Now every student listen to me. Some water molecule, see now where all the water molecule reaches, friends, this is the most important one. Some water molecule reached where? Water molecules reach the epidermis. Now some of the water molecule to enter into the xylem follow the symplastic pathway. Some of the water molecule to enter into the xylem, they will follow the apoplastic pathway. Don't confuse. Should not think that all can follow one pathway. No. Some of the water molecule can follow the symplastic, some of the water molecule can follow the apoplastic. Now every student pay attention. Whatever I am drawing in the white color, this is the movement of water molecule via symplastic pathway. You only try to understand. The water molecule is moving from epidermis to the cortex, cortex to the endodermis, endodermis to the pericycle. Finally, they entered into the xylem. So what I would like to conclude here, I would like to conclude whatever the water molecule that follow the symplastic pathway from the epidermis, they will directly enter into the xylem without any disturbance within the cell. What friends? Inside the cell, by using the plasmodesmata. But I told you all the water molecules are not following this particular symplastic pathway. As we discussed, certain water molecules are actually following the apoplastic pathway also. Now every student listen to me, I'll explain twice. Whatever the water molecule that are following the apoplastic pathway, they cross the epidermis through the cell wall. Every student look into this, through the intracellular space. It will cross the cortex through the cell wall and intercellular space. But friends, whatever the water molecules that are following the apoplastic pathway, once they reach to the endodermis, what friends? Once they reach the endodermis, do you think they will transport? Never ever. They can't transport. You can ask me why. Because on the endodermis, what is present, friends? Casparian strip is present. We all people know very well, Casparian strip never ever allow the movement of water molecules, because Casparian strip contain a chemical. We know, right? Casparian strip contain a chemical named as suberin. Suberin never ever allow the movement of any ion or any molecule inside the cell. Now friends, everyone listen. Now what happens? All the students listen, look into the arrow what I am drawing. From the endodermis, water molecules skip the apoplastic pathway. What friends? From the endodermis, water molecules skip the apoplastic pathway and they will be re-enter into the symplastic pathway. They will be re-enter into the symplastic pathway and they reaches the xylem. I will re-explain for all of you. Once again, listen to me carefully. Whomever did not understand, once again focus. This is very important, friends. First, how water molecules are reaching from the soil into the epidermis, that is the extension of the root hair, via process of osmosis. Absolutely true point, no doubt about it. So once it reaches to the epidermis, some of the water molecule to enter into the xylem uses the symplastic pathway. Some of the water molecule to enter into the phloem uses the apoplastic pathway. Every student pay attention. Whatever the water molecule that followed the symplastic pathway, they can easily cross the epidermis, cortex, endodermis and pericycle, finally reach the xylem with the help of plasmodesmata. But friends, whatever the water molecule that following the apoplastic pathway, they can cross the epidermis, they can cross the cortex. But friends, when the water molecule reaches the endodermis, they can't move further. Why they can't move further? What's the reason? Because there is a presence of the Casparian strip. Because of the presence of the Casparian strip only, friends, it never allow the movement of the ion or molecule. Then what happened, friends? From the endodermis, the water molecule will enter from the endodermis, they skip the apoplastic, they re-enter into the symplastic. Once again via plasmodesmata, they reach the xylem. These 2 points you people has to remember compulsory in the apoplastic and the symplastic pathway. Kindly don't forget the point at all. Okay, now finally water reached where, friends? Finally where the water reached? Finally water reached the xylem. Now where the xylem is present? Xylem is present in the root. Now from the root, the water molecule has to be transported into the tip of the 200 feet plant or 100 feet plant. Is it? How it will occur? What is the xylem? What does the xylem is composed of? How does the movement of water molecules are mediating via xylem? Let's try to understand elaboratively. Let's look into the structure of a xylem. Friends, once you look into the structure of a xylem, every student listen to me. Xylem contain parenchyma, xylem contain tracheary elements. Don't forget this. What the xylem contain? Xylem contain parenchyma, xylem contain tracheary elements. Everyone keep it in your mind, friends. Parenchyma, wherever you read, friends, wherever you read, common statement, keep it in your mind. Parenchyma is living tissue all the time. Tracheary elements are the dead tissue. So if I ask you a question, so xylem contains 2. So among these 2, which can conduct the water efficiently? Listen my question carefully. In the xylem, which can conduct the water efficiently? Most of the students will give the parenchyma as an answer, which is absolutely wrong statement. Parenchyma never involved in the transport of water efficiently, right? So which will do is the tracheary elements. You can ask me why? Because friends, you can see the name itself, na? Tracheary elements are dead tissues. Since they are dead, is there anything present inside? Nothing is present inside, no friends. Inside the tracheary elements, a huge space, hole, lumen, nothing is present. So my friends, as a result, it can easily transported into inside. Very, very simple point. So that's why in the xylem, tracheary elements are said to be the most important water conducting elements. Kindly don't forget it. And this particular tracheary elements consist of tracheids also, and the tracheary elements can also consist of vessel element. We people has to know the major differences between the tracheids and vessel elements compulsory. Even friends, in the year 2017 also, 2019 also, 2015, 2014, even 16, many times the question used to come from tracheids and vessel elements. So I want you people to remember every single difference between this particular tracheids and vessel elements. It's a very simple, friends. Let me define you in a simple way. One point is common, na? One point is common, whether it is the tracheid, whether it is the vessel element, doesn't matter. Both are dead in nature, no doubt about it. Whether it is the tracheid, whether it is the vessel element, it doesn't matter. Both are dead cells only. So every student listen my words very carefully, friends. All of you look into this. Let me explain in a simple way. Let's talk about first tracheids. Whatever the points that I will highlight, I want you to concentrate on that points majorly. Tracheids are the major conducting element of the xylem, especially in the gymnosperm and ferns, but not in the angiosperm. All the students look into the diagram. Very simple to remember. Tracheids are elongated spindle fiber in shape. Simple. They are elongated spindle fiber in shape. And what do they contain? They contain pits. Pits means small holes. You can call it a pit. You can also call it as a pore. Pit or pore, P-O-R-E. So it contains a pit or pore through that particular pit or pore only, friends, the water transport can occur. Once again, listen to me carefully. Tracheids are majorly present in the gymnosperms. What is the point I want you to remember, friends? They are long, elongated spindle fiber in shape. They contain lot of pits and pores, and using that particular pits and pores only, friends, they will be transporting the water. But I want you to remember, in case of a tracheids elements or in case of a tracheids, perforated plates are not present. Okay, but where the perforated plates are present on the vessel elements, especially in the angiosperm. That is the reason, friends, we can call the angiosperms or the more efficient when compared to this particular tracheids. Never forget that particular statement. Okay, tracheids are the long elongated spindle fiber present majorly in the gymnosperm, contain pits and pores but do not contain perforated plates. Every student listen a point. Since they do not contain the perforated plates, we never call tracheid is the major element for the transport of water. We never call that. Now, friends, we can call tracheids are very lower efficiency in the water conduction because no perforated plates are there. That's why they are present in the gymnosperm. Of course, gymnosperms are not highly modified when compared to the angiosperm. Next one, friends, vessel elements. Angiosperms. Angio. Friends, what is the full form of angio? Angio, vessel-like structure. Angio is what, friends? Angio is the vessel-like structure. So, angiosperms are the one where the vessel elements are present. Friends, easy to compare. We read na, tracheid elements are elongated structures, but they are very short. They are very short, but they are wider. Very simple point. They are shorter but wider. What the tracheids contain? Tracheids contain pits or pores. Friends, same way here also, where in case of perforated plates also, friends, in case of vessel elements also, pits or pores are present. But along with the pits and pores, in the vessel elements, perforated plates are also present. As I mentioned, perforated plates are more efficient in conduction or in the transport of the water. That's why we can say vessel elements which are present in the angiosperms, they are having a very higher efficiency, very higher efficiency in the water conduction. Kindly do not forget this particular statement at all. Okay, so friends, finally where the water molecules are there, finally water molecules are entered into the xylem. Now, friends, where does the xylem is present? See, this is the xylem. Xylem is present in where? In the root. Suppose this is a 100 feet tall plant. Now, from the root, how does the water molecules are moving 100 feet tall very faster? Of course, there should be some mechanism. There should be a certain force. So what is that mechanism or what is that force? We can call it cohesion tension theory. So first listen. Suppose example, just imagine what I am saying. Suppose example, I am drinking a cool drink or I am drinking a carbonated water or a soda by using a straw. Everyone imagine what I am saying. So whenever I am drinking a cool drink or a soda by using a straw, when I keep the straw in my mouth, what happens? Little air releases into the water molecule. As a result, what happened? One water molecule associated with the another water molecule. How, friends? By the process called as cohesion. At the same time, water molecules attaches to the wall of the straw. How? By the process of adhesion. Very simple. Then you can ask me, sir, how they will go up? So when we start sucking, obviously the water chain will come up. Friends, imagine the same mechanism here. Just replace the straw with the xylem. So in the xylem, one water molecule can be associated with the another water molecule like this. By what mechanism? Cohesion. How, friends? This is one water molecule. This is another water molecule. So what is the linkage, friends? OH. So like this, one water molecule can be associated with the another water molecule. We call cohesion. The next one, adhesion. All the water molecules are attached to the wall of the xylem. Now water chain is ready. How does the water chain go up? The water chain is going up because of the transpiration pull. Because of a tension, we call it transpiration pull. How does the transpiration pull occur? How does the movement will occur? We will read in the tomorrow class. Okay, so tomorrow I will explain the very clearly about the xylem movement, phloem movement. Immediately, friends, we will try to finish the little left part of the secondary metabolites also. Right, by tomorrow the secondary metabolites also will be completed. Okay, is it clear, friends, everyone, till this slide? We will continue once again from the cohesion tension theory onwards in the tomorrow class. Hope it's clear for everyone. Okay, that's great. Okay, if you have any doubts, you can ask me. So kindly attend at 8:30 a.m. class without fail. So we will finish the endocrine system. We will move on to the respiratory system. Don't forget. Thank you. Thank you all. I'll see you in the 8:30 a.m. class, friends. Bye-bye. I'll see you at 8:30 a.m. Thank you all. Thank you so much. Bye.